Okay, our next unit is genetics. Gregor Mendel is considered the father of modern genetics. So homologous chromosomes. Remember, homo means the same, so homologous chromosomes are going to be the same. They are paired chromosomes with genes for the same traits arranged in the same order. That means if there's a gene for a P hat here, there's going to be a gene for the P hat in the same place in the homologous chromosome. If there's a gene for leaf hairs, there's going to be a gene for leaf hairs in the same place, and a gene for the stipple shape in the same place. They're homologous, they're the same. One came from the mama plant, one came from the daddy plant. And so the homologous chromosomes must match in physical size. They're going to be the same size, and they're going to be the same location of the genes and have the same type genes. So genetics is the study of how traits are passed from parent to offspring. The traits of the parents are passed from offspring by sex cells, which are also called gametes, which are also called germ cells. And we know these in the male as sperm and in the female as the egg. So sex cells, gametes, germ cells, sperm, egg, they all mean the same thing. So sex cells, it has one of each kind of chromosome present. Okay. You might get a chromosome for height from dad and a chromosome from height from mom, but you'll have one from each parent. They have half as many chromosomes as the somatic cells. Remember, somatic cell means body cell. That's just like your muscle cell, your heart cell, your skin cell. Okay. They're going to have, somatic cells are going to have twice as many. They're going to have the full 46 chromosomes. Sex cells are only going to have 23 because you're going to get 23 in that sperm from dad and 23 in that egg from mom. And they're going to come together to make the full 46 for all your other cells. Sperm in the male, egg in the female. Sex determination. Sex chromosomes are chromosomes that determine your sex. Okay, we have 23 pair, and that last pair is what we call the sex determining chromosomes, the XX or the XY. If it's a female, it has two X's, a male has an X and a Y. Okay, so what this means is mama can only give an X chromosome to a baby. One of her egg cells will get this X, and the other egg cell will get that X. Okay. Dad, when he goes through meiosis and makes sperm cells, half the sperm cells will have an X, and half his sperm cells will have Y. So it's going to be dad who determines if everybody's going to get an X or mom. So dad's going to determine if the baby gets an X or a Y, and he's going to determine the gender of the baby, if it's going to be a girl or a boy. Oops. Sorry about that. Here we go. Okay, here we go. Females. Females have only one kind of sex chromosome. They can only make one kind of egg. Each egg of the female has one X chromosome. Males have two different chromosomes. They can make two different kinds of sperm. Each sperm of the male will have either an X, half are going to have X, half are going to have Y. So offspring receives an X chromosome from the mother, and it's either going to get an X or a Y from the father. So when it determines the sex of the child, it's going to be the father. So ladies, don't let those men, when you get married, when you're in your way up in your 30s and you start having babies, don't let the daddy fuss at you because you keep having girls and he wants a boy. He's the one that determines it. It's up to the dad's sperm. The X or the Y comes from the dad. So the child receives the father's X chromosome, the child will be a girl. If the child receives the Y chromosome, the child will be a boy. Sometimes, once the egg is fertilized, sometimes it can split, and that'll make twins. Identical twins, if the egg has already been fertilized with the sperm, and then that one cell, or couple of cells, maybe it's already divided a couple of times, but that will make identical twins like me and my sister. Fraternal twins means two totally separate eggs fertilized by two totally separate sperm. That would be fraternal twins. Okay. So identical twins are formed from one fertilized egg. They have the same sex, of course, because they came from the exact same egg and sperm. Okay. They will have identical DNA, identical twins, identical DNA. Fraternal twins are formed from 2 or more fertilized eggs. They may be the same gender or different genders. You can have boy and girl twins that are fraternal twins, because maybe this egg was fertilized with the X sperm and this egg was fertilized with the Y sperm, and you can have boy-girl fraternal twins. They have different DNA. They're not any more genetically alike than normal brothers and sisters. So meiosis. Meiosis is how we make those germ cells, those sex cells. So it's a kind of cell reproduction that forms egg or sperm. Before meiosis begins, each chromosome duplicates and forms a sister chromatid. And so they're going to copy. They start out as just a single, one strand, that single side of the X is their chromosome. And it's going to duplicate. They're stuck together in little X's. That's called sister chromatids. And it's going to be much like mitosis, except we're going to go through it twice. So we have prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. PMAT. Remember, PMAT, prophase, metaphase, anaphase, telophase. And then cytokinesis. And then we do it again. Prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. PMAT again. PMAT 1 and PMAT 2. So prophase 1, the sister chromatids shorten and thicken. That's when we begin to see the little X's that we can actually see under the microscope. And the nuclear membrane begins to break down. Much like prophase in mitosis that we learned about earlier. So matching chromosomes come together to form pairs. Where we have the chromosomes line up single file, in mitosis, they're going to form pairs called homologous pairs. The homologous chromosomes are going to line up. So you're going to have, maybe this was chromosome 1 from dad, and this was maybe chromosome 1 from mom. And they're going to line up in homologous pairs. And you're going to have chromosome 2 from dad, line up with chromosome 2 from mom. So they're going to line up in homologous pairs. And we're going to call this a tetrad because we actually have 1, 2, 3, 4 copies of the chromosome together there. And then 1, 2, 3, 4 copies of the chromosome there. That's a tetrad. Tetra means 4. So we call these tetrads when they line up in homologous pairs. So these pairs may exchange a portion of the chromatids. In the process, this is called crossing over. Okay, so right here, with these we may get something that looks like that. Maybe we'll just use a different color to represent the other one. They're going to kind of cross over each other. So I call it crossing over. And then it may come out looking something like something like that, where part of this chromosome is going to this one, and part of this chromosome is going to this one. This gives us more genetic diversity, and genetic diversity is very, very, very important in a population. Okay? Like we're going through right now, we're going through something that comes in, and a lot of people are susceptible to it, and a lot of people start dying from it. Well, if we have some genetic diversity, there may be somebody that has an immunity to it, and that may allow the population to carry on. So genetic diversity is very important. Crossing over, this crossing over that happens here, crossing over is very important in increasing genetic diversity in a population. That's what makes you not look exactly like your siblings. So these pairs exchange portions of the chromatids in a process called crossing over. Crossing over is what makes 6 cells unique and why you're similar to your siblings but not identical. And the centrioles begin to move to the opposite ends of the cell, just like they did in mitosis. So here we go, prophase 1. We have our homologous pairs. They're crossing over. Okay. Homologous pairs. Those are homologous. These are homologous. Meta means middle. Meta means middle. And again, we're going to line up down the middle. But our homologous pairs, it's not a straight line like in mitosis. They're lined up in their homologous pairs. Okay. This is going to be a big difference from mitosis. In meiosis, we're going to line up in pairs, because during our first anaphase, we're going to pull these whole sets of X's apart. Okay? So centrioles have moved to the opposite ends in metaphase, and pairs of homologous chromosomes are lined up in the center or the middle of the cell. Meta means middle. They're lined up down the middle, and they're homologous pairs for metaphase 1. The anaphase 1, the tetrads split apart. They still keep their little X's. They still have sister chromatids together, held together at their centromeres. Sister chromatids are still together, but the homologous pairs are homologous pairs pull apart from the opposite end of the cells. And then we have telophase, telophase. Telophase is where it starts pinching in two. The cell membrane begins to pinch in. Cytokinesis occurs, which divides the cytoplasm. This results in the production of two daughter cells. Each daughter cell now contains one chromosome pair that was in the parent cell. So now we have one of each chromosome. Prophase 2, we're going back to prophase. Not a lot happens here because they're already condensed into the little X's, and it goes really, really quick. Sometimes it's passed over entirely. So now we have just one of each chromosome. It may be a mama and a daddy, or maybe a daddy and two daddy cells, or two daddy chromosomes in the cell, because they separate randomly. So sister chromatids are going to line up across the middle again. Meta means middle. Metaphase 2, this looks more like mitosis this time because we just have our single X's lined up down the middle. Centrioles are at the opposite ends again. This time, anaphase 2, this is where we're going to separate our sister chromatids. Our sister chromatids are going to pull apart and go to the opposite ends. So sister chromatids are pulled apart. Notice we have two cells going through this now. At the end of T1, we had two cells. Now these two cells together are going through PMAT2. So the sister chromatids are pulled apart to the opposite end. Sometimes they don't pull apart like they should. Sometimes they may stick together and an egg or a sperm may end up with two copies of a chromosome. And the other one would end up with no copies of that chromosome. And failure of homologous chromosomes to separate properly in a gamete ends up with too many or too few. And this is non-disjunction. And one non-disjunction case that many of us are familiar with is Down syndrome. And with Down syndrome, a person would have an extra chromosome number 21. And that would mean instead of these pulling apart, maybe they got stuck together in this egg cell. And then when that egg cell got fertilized, because that means they got stuck together, so it still had two. And then when it got fertilized with the sperm cell, which brought in another chromosome 21, then you end up with what's called trisomy or 3 chromosome 21s. Non-disjunction, important word to know. Non-disjunction can result in mutation of a cell having too many or too few chromosomes. And most of the time, it's going to be too many chromosomes. Very rarely does a full monosomy survive. Okay, so most of the time, it's going to end up being a trisomy, or sometimes even more than that. Non-disjunction ends up with too many or too few chromosomes. Telophase 2, the nuclear membrane begins to reform around each new set of chromosomes. Cell membranes pinch in, cytokinesis occurs, and the cytoplasm is divided in half. Remember, we have this going on with two cells now because at the end of PMAT1 we ended up with two cells. So now at the end of PMAT2, 4 new cells are formed. These 4 new cells are called germ cells, gametes, sex cell, sperm, or egg. They are haploid in number, which means they have half as many chromosomes. So telophase 2, pinch it in, and we end up with 4 totally different egg or sperm cells at the end of cytokinesis of meiosis. So meiosis, here's a nice picture of it. Prophase 1, metaphase 1, line up in homologous pairs. The full sister chromatids together, the little X's still pull apart. So we end up with 2. Then we go through prophase, metaphase, anaphase, telophase. 2, and we end up with 4 totally different, genetically diverse cells. And that's what it would look like under the microscope. You can see middle, right there. Since meiosis is a process by which gametes are made, it's sometimes called gametogenesis. And males' gametes are called spermatozoan, we call them sperm for short. And the process is called spermatogenesis. In females, if the egg cell ovum are produced, it's called oogenesis. Ovum is just another word for egg. It's called oogenesis. Once the gametes are formed in the parent organisms, they must be fused during fertilization in order for sexual reproduction to occur. Each parent contributes 1 haploid gamete. This produces a new organism that is diploid in number, and the result is termed a zygote. So once the egg and sperm come together, the egg is fertilized by the sperm cell. It's once again diploid or diploid. 23 from mom, 23 from dad come together and it makes what's called a zygote. That moment of fertilization when it goes back to the full chromosome. It's a zygote. The zygote begins to grow by cell division through mitosis. So now the somatic cells are all fighting through mitosis to make the embryo that develops into a fetus and then is delivered as a baby. Very precious. Okay, mitosis and meiosis, the difference. Mitosis, we end up with two identical cells. Meiosis, we go through PMAT twice and we end up with 4 genetically different cells. Those are our gametes. This is happening, mitosis is happening in somatic cells. Meiosis is forming gametes or sex cells. So the differences between mitosis and meiosis. Mitosis makes somatic cells or body cells. Remember, somatic means body cells. It is with 2 cells that are identical to each other. So if you're taking a test, you see 2 identical cells, then it went through mitosis. If you're taking a test, you see 4 unique cells. Remember, they're unique, they're crossing over, they swap some genetic material back and forth. So 4 different unique cells are crossing over. That was meiosis, making germ cells or gametes. Okay. Asexual reproduction. That's asexual reproduction is reproduction without a mate. That's what bacteria do. They just go through what's called binary fission. They copy their DNA and they split in 2. So disadvantages is offspring are genetically identical to the parent. That means that all of a sudden something changes in their environment and they're susceptible. That means every single one is susceptible because they're all just alike. And that can wipe out the population because there's very little genetic diversity in asexual reproduction. And the offspring respond to environmental changes in the same way. So that makes them more susceptible to the population being wiped out. Disadvantages of sexual reproduction require specialized sex organs. It has to be a more highly developed organism. Could result in offspring with mutations. Remember, we can get things like non-disjunction happening. And it requires more metabolic energy. You have to find a mate, you have to reproduce, it takes a lot longer to go through sexual reproduction. That's the biggest disadvantage of sexual reproduction. It takes a lot longer, requires a lot more energy. Big advantage of sexual reproduction. The main reason they use sexual reproduction is to get that genetic diversity, survival of the species through genetic diversity. Gregor Mendel is considered the father of modern genetics. He was an Austrian monk. He worked in his garden with pea plants. And he used those pea plants to study dominant and recessive traits. We know now that chromosomes contain genes, and genes are those little codes for our proteins that control our eye color, our hair color, our body shape, and all those things. So a gene is a small section of a chromosome that determines certain traits. And you get some examples of traits: eye color, height, the shape of your nose is even a genetic trait. Size of your ears. So chromosomes in the body are paired. The alleles are things that determine how genes are expressed on chromosomes in somatic cells. They are paired also. 2 alleles representing a gene make a pair, and this is a combination of alleles that determine a trait. For example, traits such as eye color, nose shape, we use upper and lowercase letters to represent our alleles. Okay, we usually use an uppercase letter to show that an allele is dominant. We use a lowercase to show that an allele is recessive. One allele comes from mother, and one allele comes from the father. In a monohybrid cross, Mendel looked at several traits in pea plants. One of those traits was the height of the pea plant. He noticed that tall parent plants made with a short parent plant, they all came out tall. Okay, so they all came out with the tall trait because tall is dominant. If you end up with a tall parent and a short parent, and we get a tall gene from one parent and a short gene from the other parent, we get dominant tall, recessive short. If that dominant trait is there, even if the recessive trait's there, if that dominant trait's there, that dominant trait will always show up. So big T is tall, little t is short. And the example is always in genetics. The uppercase letter represents the dominant allele, the lowercase represents the recessive allele. To determine which allele the offspring would have, we do what's called a mono, meaning one. Monohybrid cross, we're going to look at one trait. we might take what we call purebred tall, monohybrid trait, Punnett squares. You'll probably remember these from middle school. Okay. If it's purebred tall or homozygous tall, it would have two big T's. If it's homozygous or purebred short, it would have two little T's. To be short, it would have to have two little T's because that's a recessive trait. So short would have to be little t. Tall, if it's homozygous, big T, or like the example I showed you just a second ago, even if it was big T, little t, it would still be tall. But here we're saying it's a homozygous tall crossed with a homozygous short. So we have big T, little t, big T, little t, big T, little t, big T, little t. So all of these offspring would have the big T, little t, which is called hetero. Hetero means different. They have two different alleles. Heterozygous. There's a dominant allele and a recessive allele, but because that dominant allele is there, they would be heterozygous tall. Okay. If you cross two of these offspring, they're both heterozygous for the tall trait. They're both big T, little t, and big T, little t. So this one we get a big T from mom and a big T from dad. A big T here, a little t here, a big T here, a little t here, a little t from here, and a little t from here. Okay, so this one, this is a big T, big T. This would be homozygous. That would be homozygous tall. And that's 25%. One out of those four is homozygous tall. We have 2 out of 4 that are big T, little t. They have different traits, so we call it heterozygous. That would be heterozygous tall. And we have 2 out of our 4, so that's 50%. And then we have one, this little t, little t. This will be a short plant. Short trait. We could say homozygous, but if it's showing that recessive trait, it has to be homozygous. So homozygous short, and it would be 25%. And that is a monohybrid cross. Looking at one trait. Okay, so some of the pig plants were purebred, had the same big T, or little t. Big purebred, homozygous, it's the same thing. So if two parents are crossed, that's what we just did, the homozygous tall with the homozygous short. Here's a Punnett square example. If we have a heterozygous, big A, little a, we get one heterozygous, actually we get two heterozygous showing the dominant trait, and then we get 2 that are showing the recessive trait. This is maybe brown eyes and blue eyes. So maybe dad has blue eyes, which is a recessive trait, so he has 2 little b's. Mom has brown eyes, but carries the trait for blue eyes. Then you would end up with 50% offspring with the brown eye trait, and then 50% offspring, heterozygous for the recessive blue eye trait. So alleles keep other alleles from showing up. They are dominant. They do not show that trait. If the heterozygous condition, you don't see that trait, then that trait is recessive. An organism with 2 dominant traits is said to be homozygous. Homo means the same, or purebred dominant, such as brown eyes is big B. An organism with 2 recessive traits, have the recessive trait is homozygous or purebred for the recessive trait. These alleles will be written in lowercase, such as blue eyes or little b, little b. An organism with 1 dominant and 1 recessive trait, such as big B, little b, is heterozygous, or also called hybrid. Okay. Hybrid would mean it has a gene from, so it could be heterozygous or hybrid. It means the same thing. It means it's going to have a dominant gene and a recessive gene. Of course, the dominant gene is going to show up. It's going to show that dominant trait. So are they following homozygous, dominant, homozygous, recessive, or heterozygous? Okay. big G, little g. It's a big G, little g. What do you think? There are two different ones, so they're going to be heterozygous. And it's going to show that dominant trait. Okay. Big H, they're the same, so it's homozygous. And it's going to show that dominant trait. Little b, they're the same, so it's homozygous. And this time they're both little letters, so it's going to show that recessive trait. Big S again, homozygous, shows the dominant trait. Big K, little k, they're different. Heterozygous, but it does have the dominant allele, so it shows that dominant trait. Again, big R, little r, they're different. Heterozygous, has the dominant allele, shows the dominant trait. Little a, homozygous for the recessive trait. BQ, homozygous for that dominant trait. Original Punnett came up with the idea for the Punnett square. That's those little squares that we used a minute ago that you can go through and determine what alleles can go to the gametes when they're formed. So you draw your Punnett square, and each square is going to stand for each possible allele, or each possible gamete that could come out. Decide what your allele combinations are for your parents. Then you're going to write those in there. For example, if the mother's egg cells alleles are big B, little b, and the eye color trait we were discussing, brown eyes are dominant over blue eyes, so big B would represent brown eyes, and blue eyes would be the recessive little b, little b. And we're going to write those letters in. Bring the letters across the top. The boxes show what combinations are possible. We may get big B, homozygous dominant, heterozygous dominant, or recessive. So there's only one way to have blue eyes, and that is little b, little b. And that's it for today. Thank you.